Okay. Well, thank you for joining in on my webinar here. My name is Michael. I want to talk about transcript assembly and RNA sequencing. My specialty really is in developing both molecular and bioinformatic protocols for getting the best picture possible of RNA at the isoform level. And so I want to thank LEXOGEN for inviting me to perform this webinar, and it will be sort of in two parts. First, I'd like to talk about how we got to the present day, sort of RNA sequencing technologies up to now, and then I'd like to introduce my own software package I've been developing and sort of the ideas behind it and how it performs. So here is an end-to-end rethinking of transcript assembly. And in order to think about this problem properly, it's important to consider what we're even trying to look for when we are isolating and sequencing RNA molecules. And if you think about the structure of a gene, then very simply, it's a region of DNA in an organism where an RNA polymerase binds to a transcription start site. It will continue through the body of the gene, terminate at a cleavage and polyadenylation site, and then that RNA molecule receives a poly A tail and gets exported to function in the cytoplasm. However, when this RNA molecule is matured, there are many different possible ways for that maturation to occur. Most eukaryotic genes are spliced. They contain introns that actually must be removed from the primary sequence of the RNA molecule before that RNA is mature and has a continuous coding sequence. In addition to constitutive splicing, many eukaryotic genes, in fact, have multiple alternative ways that their introns can be spliced. For instance, we see an example here, just a schematic example of exon skipping, where you have an exon that may normally be included in the mature transcript, but is bypassed by the splicing machinery in an alternative isoform. Additionally, you can have introns that sometimes are retained within the mature sequence. On top of that, the donor and acceptor sites are not always fixed in one position, and they can actually vary and cause some microsite variation inside these RNA molecules. And on top of all of this splicing variation, there's also the possibility for many genes in the genome, in fact, to either initiate or terminate at positions other than the canonical transcription start site or polyadenylation site. And so when you add up all of this variation together, you actually get an immense potential for RNA variation, even from one single eukaryotic gene. And so that begs the question, if we're interested in isoform variation, how do we go about experimentally identifying transcript isoforms? And I thought it would be fun to go kind of all the way back and think about how things were done back in the old days. So the concept of sequencing really came from Fred Sanger and colleagues in the late 70s. And in the method that Sanger developed, one clone of DNA was replicated many times with a DNA polymerase, but with the caveat that in each of the 4 nucleotides, he included different chain terminating inhibitors. So these are nucleotide analogs that can be incorporated by the polymerase, but actually stall the polymerase reaction. And so with these chain terminating inhibitors at the positions of G's, A's, T's, and C's, it was possible to build up a library of clones that were progressively shorter by 1 nucleotide each. And then you could read back the progressive length of these terminated molecules and in essence, sequence each of those nucleotides in order. The fundamental science of Sanger sequencing, as it eventually was called, improved over the years, and these chain terminating inhibitors these nucleotide analogs were swapped out for fluorescent analogs, and it was found that these reactions could essentially all be performed in the same, or they could be combined. And so then the 4 colored fluorescent reporters for the 4 nucleotides could be read off in a much more rapid way than running the whole thing on a gel and looking at it by eye. And so as the technology of Sanger sequencing improved, the throughput of what could be sequenced also improved. And eventually that brings us to what's often called the genomic era. And so around the beginning of the 21st century, there were a collection of very large-scale sequencing projects where genomes of different plants and animals were selected, and chunks were cloned and Sanger sequenced. This was also in combination with an early form of shotgun sequencing. But here around the early 2000s, we start to enter an era where we have hundreds of thousands or even millions of DNA fragments. And using reverse transcription, then actually RNA could be converted to cDNA and sequenced as a collection of hundreds of thousands of clones. And these clone collections could then be used to annotate what transcripts are derived from the genome. And with the advent of these large cDNA libraries, actually came the need to develop algorithms that compressed this information and sort of summarized it in a way that was useful and representative of the transcript isoforms. So this is one of the first what would be called transcript assembly algorithms. It's called PESA, and it was developed just after sort of the sequencing of the Arabidopsis genome in order to actually annotate the transcript isoforms that came from the genome. And it used a form of overlap graph that we'll see coming back quite soon. But only a few years later, the technology developed where it was possible to, instead of sequencing, say, hundreds of nucleotides from a single clone, to develop a highly multiplexed library of short cDNA reads. And these short reads could then be sequenced in parallel and developed and produce millions or tens of millions of sequencing reads in what's called RNA sequencing. These platforms, eventually what today we still use the Illumina short read sequencing platforms for. We can collect millions of short read fragments and then map them to the genome, and they can either map along the body of a read, or they can map with a gap, and that gap reflects the intron that was spliced out of the mature molecule. So with millions of short reads, of course, that necessitates new algorithms to piece them back together. And one of the most influential is Cole-Trapnell's cufflinks method. And cufflinks was a transcript assembler that took these short read fragments, built them into an overlap graph, and then found the most parsimonious set of paths through this graph that could reconstruct essentially the best possible guess or the best explanation for the reads that mapped to the transcriptome. The algorithms got faster, more powerful, and more capable of handling a larger total number of reads. And in 2015, StringTie came along and sort of reimagined, instead of an overlap graph, a much more condensed representation that the authors called a splice graph. The splice graph was much faster and lightweight and lighter weight to work with, and they were able to generate with more total reads, larger, work with more total data and generate assemblies from that. But that actually causes us to run into a pretty big barrier with transcript assemblers, and that's how can you actually know that an assembly is correct? One method that was quite popular and still is to use simulated data. And so the authors of StringTie are showing here, essentially a collection of simulated short reads from the reference transcriptome that try to emulate what an RNA sequencing experiment would look like. And they can, from there, use a ground truth. They know what was simulated, and they can see how well the algorithms reconstructed. But unfortunately, simulation is not reality, and there are a lot of things that happen in the actual chemistry of reverse transcription, of generating the sequencing library, that introduce different biases and errors that you can't fully account for through simulation. Another possibility for benchmarking these algorithms is to simply assume that the reference annotations are correct and complete. And you can come up with metrics for precision and sensitivity as a function of how well your assembly matches a reference annotation. But obviously, you wouldn't need to do any assembly at all if the reference annotations were correct and complete. So both of these assumptions are known to be false. And the incompleteness of the reference annotations and the possibility that they actually contain errors are both reasons to justify doing transcript assembly. And so, these challenges, these problems led people to search for ways to introduce a ground truth to the sequencing experiment. One of the more influential techniques was the idea of synthetic spike-in RNA molecules. This set of ERCC spike-ins that was published over a decade ago now has been incorporated into a lot of sequencing libraries, and it's a set of what was 96, now it's 92 distinct synthetic RNA molecules that can be spiked in a known concentration into your sample that you're sequencing, and you can understand essentially how quantitative your RNA sequencing method is, or your quantification method is. This in itself was extremely useful to know, is whether or not the RNA molecules we counted were proportional, whether or not we could say anything about the quantity, the relative quantity of RNA molecules from a sequencing experiment. But qualitatively, these spike-ins are actually quite simple. And we can see, I'm just showing an example of 4 ERCC sequenced samples on 4 of the 92 total transcripts. And they're short, simple, linear molecules. And they were really designed for testing quantitative accuracy, but they don't reflect the real complexity of real transcripts. So if we go back to the isoform complexity that I showed in the sort of hypothetical diagram at the beginning, this isn't merely a hypothetical. Here is an example of a real human gene that's been annotated, and you can see a massive variety of alternative transcripts produced from this one gene. In fact, the whole suite of possible transcript variations can exist at any given gene in any genome. And so it really is necessary to have a ground truth that reflects that amount of variation. And so, as you'll see later, LEXOGEN has produced a collection of synthetic RNA spike-in molecules that are designed to emulate various isoforms across seven synthetic genes. I'm showing how the structure of the SERV3 isoforms look on the synthetic genome. And you can see that these molecules are designed to emulate different transcript variation types, such as exon skipping and intron retention. They also have alternative transcription start sites and polyadenylation sites in the synthetic molecules, as well as a collection of antisense RNAs, which can really have a negative impact on both annotation and quantification if you're not very careful. So a more complex sort of ground truth included in a sequencing experiment can go a long way to informing you how correct you are. And that brings us to the RNA sequencing technology that's available today. There are other ways to validate different aspects of your assembly. So most assemblers were designed for short-read sequencing, and we can see that shown here as little fragments of the total RNA pool. However, more sequencing techniques have come along, including methods that selectively capture and amplify the precise RNA 5' end, the transcript start sites, or the 3' ends, the polyadenylation sites of molecules. And moreover, in the last 6 to 8 years, these third-generation sequencing technologies have come along, including PacBio and their isoforms platform, or Oxford Nanopore Technologies. And these two methods can actually sequence long reads from one end of the molecule to the other. And even though they're much lower throughput right now than short read technologies, the ability to sequence the entire structure of an RNA molecule from end to end is extremely valuable in knowing the total structure of that RNA. And in parallel to these developments, there's actually been an astounding amount of miniaturization for these methods in the form of single-cell sequencing technologies. So I can't go into all of the methods that have been developed, but in recent years, single-cell sequencing has really taken off, and we now have many different methods to isolate RNA from single cells, barcode them, and then sequence them as a large library without losing the information of where each RNA came from, which cell each RNA came from. And so that leaves us in a very interesting position, because we have many different methods to capture RNA molecules, but it isn't totally resolved what actually is the best way to identify transcript isoforms from cells of interest. And so I hopefully can give you what I believe is a good answer to that question through the software that I've been developing.